I'm going to call up the document itself. You should be able to see it now, can you? Cystic fibrosis mutant.
Yes.
Try to make it a little bit bigger. And we'll skip down past the mutants to your CF project worth up to 50 points per student. Now, since you saw it previously, I've changed the time frame from 10 minutes to 8. And that's because it turns out there are 10 groups. And with 8 minutes per group, that's 80 minutes, and a class period is 75 minutes. So that's close enough. And so tentatively, it's still planned for May 26th. You'll make your presentation. And prior to making your presentation, in other words, prior to May 26th, you'll need to have me review what you have. So the times that you can schedule, and I'll send you a memo about this, so you'll have it in black and white. I'll just tell you for right now that May 13 and 14, that's a Thursday and a Friday, and 20 and 21, that's a Thursday and a Friday a week later. On those days, anytime, all day, you can schedule a 30-minute time period with me to review your, yes?
I have a quick question on that. So do the whole group have to be making appointment with you, or just one person in the group?
The whole group. You'll review it as though you were presenting it to the class.
Okay, thank you.
Okay, and so what I'd like you to do is send me an email telling me your preferred time slot, and that would be on any of those 4 days for a 30-minute period. So when you get your group together, you can decide when is the best time for you to do a review. And then when I agree with the suggestion you've made, and I'll have to make sure there are no conflicts, then you will send me a Zoom invitation. And I'll accept that, and we'll have a review.
No one ever asked me to make chicken for them because
Yeah? Could you unmute yourself? Okay, I don't see or hear anyone.
I have a question. So basically we gonna choose the group, or you are going to choose it?
I have already chosen it, and that's next on the agenda. I'm going to stop. I sent you the groups and the mutant assignments. You should have that in your Canvas box, but apparently if you haven't seen it yet, I'll show it to you right now. Can you see that? Group members mutant?
Yes.
Okay, so this is what is in your box, your Canvas box, and you can look at it on your own. But these are the groups, numbered 1 to 10. These are the members of each group and the mutant that has been assigned to your group. So what you'll need to do is get together with the other members of your group, either by email or Zoom or whatever other means you wish, and decide how you're going to handle your project. So Everybody had a good look. Can everybody see what group they're in?
Yes.
Okay.
Yes.
All right, then I am going to stop sharing this and go back to the other one. Oh, can you see the first document again? You see
Yes.
the project, all right. I hope that's big enough for everybody to read. So the timing, and this is one of the reasons for the preview with me, is to get your timing down. I want you to be as close to 8 minutes as possible. If you're significantly over or significantly under, I'll take points off. And if you fail to have your project reviewed in advance, that's a 10-point deduction at the beginning of your presentation. Okay, so the 4 points that you need to address in your presentation are the structure of the protein that your mutant makes, the function of the protein that your mutant makes. In other words, it will have an aberrant function, and we want you to describe that. The symptoms of patients who have your mutation, and possible treatments for the symptoms caused by your mutation. And different mutations have different types of possible treatments. Then you will be graded on the 26th when you make your presentation as for how well you adhere to these instructions, the quality of your slides, and here's a link to some suggestions about how to make slides. And at the end of this document, I have a few more suggestions that I'll go over when we get there. The quality of your oral presentation, and you'll need to practice in advance speaking through your slide or slides. And finally, on the conclusion that you come to based on the evidence that you present about your mutant. Now I have in bold here that all members of each group should be involved in gathering information and organizing it so that it can be presented in a systematic fashion. Each member of the group should compose at least one slide for your group. I'd prefer more than one per person, but at least one. And the reason for that is that many of you probably already know how to make PowerPoint slides, but just in case you don't, I'd like to make sure that before you leave this class or leave this project, you know how to make a PowerPoint slide. Now, when it comes to making the presentation itself, 1, 2, or all 3 members can be involved. In other words, you can designate one person to do it all, or you can divide it up, depending on a decision that you yourself make within your group. Then, after your presentation on the 26th, I will assign a score to your presentation, and all the members of your group will receive that same score. And as I said earlier, you can get started communicating right away now that you know who's in your group and you know what your mutant is. You can start communicating and getting material together to make your presentation. Okay, any questions so far about how you're going to proceed? At the bottom of this document, I have some tips for slide making. And overall, the message is keep it simple. Pare down the content and refine your message and have one key point per slide. Increasing the messages on a slide from 1 to 2 increases the audience comprehension time by 5-fold. So it's important, if you want your audience to comprehend what you're saying, to keep the message simple and to just one per slide at the most. Simplify statements. Use the fewest words possible to make your point. And one way to go about doing that is to turn sentences into bullets. And if you have something supplemental to the content that you want to say, you don't necessarily have to show it on the slide. That can be a talking point as an aside as you discuss the slide itself. So here are some examples of how you can simplify content and decorate your slides. So the elements of style, the things that you can vary are the layout, how you, the positions on the slide that you put various things, the colors that you use, and the fonts that you use. So as an example, here is some content about a market opportunity, complete sentences and very detailed. Now that can be simplified to what's shown below, or it's boiled down to just 4 points. That itself is pretty mundane looking. It's not very interesting. So you can take that further and, for example, come up with something like this, where now you've changed the layout, made it a more interesting layout. You've added some decorations and some colors. So if you compare that, has the same information as this, but this is much less attractive, much less interesting than if you just change a few things. You can often illustrate with a picture, and here are some examples. They're advertising watches, a picture of a watch, wallets, picture of wallets, and shoes with shoes. And there are sometimes options where it's just your personal opinion. So for example, here are 2 ways of saying the same thing. One of these may be more pleasing to your eye than the other, and that's the one you should choose. And finally, when you're done with any given slide, you need to align and position the items on that slide. This is fairly easily done in PowerPoint with the alignment tool. You can align objects on the slide both vertically and horizontally. And once they're aligned, you can select the ones that you want to be held together and group them, again with the grouping tool that, that PowerPoint has. Any questions about how to proceed or how to go about making a slide? Okay.
I have a question, Professor Kiyomi Anderson.
Okay.
Is it okay to like do Google Slides because it's easier to collaborate, or would you prefer us to use like Microsoft?
I would like some level of uniformity. If you can use Google and do the same thing that you would do in PowerPoint, that's fine. As long as, Yeah, as long as there's some uniformity among the different groups.
Okay, thanks. Just wondering.
Okay.
Hello, Professor. I have a question too. My name is Zara. And can you show us where is it located? Like where is the power, like the documents that you already show it to us? Because I'm trying to find it in the Canvas, but I can't.
It's in, if you go to your home page on the global navigation, click on home at the top, and then go to the bottom where a module. So there should be, I think, 11 modules there. And in module 10, you'll see both of these documents. This one as well
Oh,
as the group of the document with the groups and the mutant assignments.
okay.
Are you looking at it now? You should be able to see that.
Oh, yeah, I saw it. Thank you.
Okay. Anything else? If you have any questions that come up after the class, just send me an email and I'd be happy to try to clarify anything else. So if we're done talking about the cystic fibrosis project, I'd like to switch to the topic of the day, which is Mendelian inheritance. You should see Mendelian inheritance. Yes, anybody can't see that?
Yes.
Okay. Now we went over much of this the last time, and I'm not going to do that again, except for a couple of points that I want to really emphasize. And the first point is, what was the state of affairs in genetics and inheritance prior to Mendel? Well, prior to that time, many people did crosses. They take parent 1 and cross it with parent 2. And what they would get is a smear of different phenotypes. Nothing that made any sense that would say anything about the mechanism of inheritance. And the reason for that is that just by taking an individual and crossing it with another individual, you're studying polygenic inheritance. And polygenic inheritance doesn't make any sense until you understand monogenic inheritance. And that's where the genius of Mendel comes into play. For the two reasons shown in red on the left, Mendel came up for the first time the idea of inbreeding, making all loci homozygous before you start. The second thing that he did that was novel is an F2 analysis. So instead of stopping, crossing two parents, looking at those progeny and trying to figure something out, he said, okay, let's cross the progeny and get more progeny. And that's when he revealed the laws of inheritance, which we'll talk about later in the presentation. But these are the two things I want you to remember about Mendel. He was the first to do inbreeding. He was the first to analyze the F2 generation. So key step 1, develop inbreds. And how is that done? Inbreds are the mating of two closely related individuals. And if you do that in successive generations in inbreeding, and in every generation you take the progeny, and again, take progeny 1 and cross it with progeny 2, go to the next generation, do the same thing. You do that 6 or more generations, By that time, all the loci in your organism are homozygous. That is, there's no allelic variation. So that when you do a cross between two different traits, those two traits are the only things that are segregating. And key step number 2 is the F2. Mendel's very first cross that he reported was between a plant with purple flowers and one with white. Now remember, these are so-called true breeding parents, so they're homozygous at all loci except for the one for flower color. So when he crossed purple and white in the F1, he didn't get a smear of variation shades of purple, variation shades of white. He got one discrete color. All the plants were exactly alike. They all had the same shade of purple, but white had disappeared. So then he took 2 purple plants, 2 of these purple progeny, and he crossed them with each other. That's what generated the F2. And now, voila, white appeared along with purple. And the ratio for every white plant, there were 3 purple ones. And so this, for the first time, identified discrete categories of progeny that were reflecting something. That making crosses was not just mixing up the contents of the 2 parents. It's actually dissecting out the characteristics of the parents. And so let me go over this. We went over it the last time, but just to emphasize how what's behind this cross that Mendel made. So the plants, he starts out with these purple plants and white plants, and he's inbred them before starting with his crosses. And so if we give purple the gene symbol big P, then we know that both big P, that both alleles at the flower color locus are big P in the purple plants. And correspondingly, we know that both alleles at in the white plant are little p. So that the only gametes that the purple plants can make are big P. And the only gametes that the white plant can make are little p. So if you cross big P and little p, the only possible genotype is heterozygous. Big P, little p. Now these progeny have an option. Since they're heterozygous, half the gametes that they make are going to be big P, and the other half are going to be little p. So that if you look at all the combinations of fertilization that can occur, as shown here in this box, which is known as a Punnett square. So if a sperm that is big P fertilizes an egg that is big P, then there will be a homozygous dominant progeny, and that will be purple. If, on the other hand, a sperm that is little p fertilizes an egg that is big P, the progeny will be a heterozygote, big P. Little p, again, it will be purple because of the big P allele. Only if a sperm with little p fertilizes an egg that's little p will you get the homozygous recessive, which is white. So the phenotypic ratio here is 3 purple to 1 white. The genotypic ratio is 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. Is that clear how that works and why the ratio comes out the way it does?
Professor, I had a question, Leanne Metzger. The only thing I don't understand is why does that make 2 for the big P little p? Like I understand the 1 and 1, but why does it suddenly get 2 for the big P little p?
Oh, because there are 2 options. So a sperm with little p can either fertilize an egg with big P, or a sperm with big P can fertilize an egg with little p. So big P sperm, little p egg, little p sperm, big P egg. Both of those give you a heterozygote, big P, little p. Make sense?
Yes, I understand.
Okay. Anything else? Okay, let's talk for a little bit then about alleles. I've already been talking a lot about them, but alleles, by definition, are alternate forms of a gene on 2 different homologous chromosomes. And homologous chromosomes is important. So that's illustrated in its simplest form here with these 2 purple chromosomes. They're homologs of each other. And if these are from an inbred, then all the other loci on both of these chromosomes are homozygous. That is, you have the same allele on both. However, what's shown here is that they're heterozygous at the locus that controls flower color. So on one homolog, there's an allele for purple flowers, and on the other, there's an allele for white flowers. So even though these chromosomes are homologous, that means that most of the genes are identical on both. They're heterozygous at this one locus. Okay, dominant versus recessive. Mendel defined what he meant by these terms. So the characteristic that appeared as the phenotype in the F1 generation was termed the dominant characteristic. And the one that disappeared in the F1 and then reappeared in the F2 was called recessive. Those are Mendel's terms, and they've stuck with us ever since. Well, these 2 chromosomes, a pink one and a blue one, again, they're homologous, but there are 3 loci on each, and they illustrate dominance and recessiveness on homologous chromosomes. At the P locus, each chromosome has a big P. So at the P locus, it's homozygous for the dominant allele. At the A locus, there's a recessive allele on each chromosome. So at A, it's homozygous for the recessive allele. At B, there is a different allele on each chromosome. Big B on the pink one, little b on the blue one. So at this locus, it's heterozygous. Genotype versus phenotype. On the left are the genotypes. Big P is homozygous dominant. The heterozygotes are big P, and homozygous recessive are little p. Now, any cell that gets a big P makes purple flowers, whether it's homozygous dominant or heterozygous. If it gets both little p, both recessive alleles, that's the only condition where it's white. So that ratio is 3:1. The genotype ratio is 1 to 2 to 1. Okay, a monohybrid cross example. A true breeding tall pea plant. And when it says true breeding, it means it's been inbred. So it's homozygous at essentially all loci. So a tall plant is crossed with a true breeding, again an inbred, short plant. Tall is dominant to short. What will be the genotypes and phenotypes of the F1 offspring? And what are the genotypes and phenotypes of the F2? You need to pay attention to this because you have in Canvas a set of problems. I think there are 10 problems that we're going to discuss on Monday. So between now and Monday, you need to go through these and work them out for yourself. And then we're going to discuss them in class on Monday. They're not going to be graded. This is for your practice and learning how to do inheritance.
Professor, this is Bennett Taylor. Are you talking about Punnett squares?
Yeah. Yeah, the best way to get the answers to these is to make a Punnett square in every case. And I'll get to this in a minute, but before doing a Punnett square, you have to figure out what are the gametes. Once you know the gametes, then the Punnett square is fairly easy. So let's go through this simple monohybrid cross where we have a true breeding dominant tall. And since this is dominant, we're going to give it a big T for tall. And it's true breeding, it's inbred, so it's big T, big T. The only gametes that it can make are big T. The short plant is true breeding, and it's recessive, so we can give it a gene symbol of little t, and it's homozygous, so it's little t, little t. The only gametes that it can make are little t. So in the F1, we're crossing a big T and a little t. All the F1 are going to be heterozygotes, big T, little t. That's the genotype. And the phenotype of the entire F1 will be tall. The gametes here can be either big T or little t. So now you've figured out the gametes for the F2, for making the F2. So we're going to cross a big T, little t heterozygote with another big T, little t heterozygote. And so you make a Punnett square. Parent 1 and parent 2. Parent 1 can make gametes big T and little t. Parent 2 can make gametes big T and little t. So if little t fertilizes, big T of parent 2 fertilizes big T of parent 1, you have a homozygous dominant progeny, big T, big T. If a little t gamete from parent 2 fertilizes a big T from parent 1, you now have a heterozygote, big T, little t. And conversely, if it's little t from parent 1, big T from parent 2, it's a heterozygote, big T, little t. And finally, if a little t fertilizes a little t, it's a homozygous recessive, little t, little t. So the genotypes then are 1 big T, 2 heterozygous big T, and 1 little t. The phenotypes will be 3 tall and 1 short. Everybody see that? Okay, a monohybrid cross is the simplest, the least complicated of the types of crosses that you will be doing in your problem set. Now, something else that you need to know and remember, because you'll surely be asked about this at some point, is a test cross. Why would you do a test cross? What is a test cross? And how do you analyze the result? A test cross, let me just add something here. You have an individual with an unknown genotype. You don't know if it's homozygous or heterozygous. And you cross it to something that you know is homozygous recessive. So in the case of purple and white flowers, if you have one of each, you know the white one is homozygous recessive. You don't know if the purple one is homozygous dominant or heterozygous. So your test cross then is to cross these two phenotypes. Now, if your unknown is homozygous dominant, then the alleles are going to be big P from one side and little p from the other parent. And that's going to give all heterozygotes. So if the progeny are all purple, then you know that your unknown is homozygous dominant. On the other hand, if the unknown is a heterozygote, then you will have a big P and a little p coming from that parent, little p coming from the white parent. So now half the progeny will be purple and half will be white. So it's an unambiguous result. If all the progeny are the same, your unknown is homozygous dominant. If it's half and half, your unknown is a heterozygote. So that's a test cross. And as I said, make sure you know when to use it and what it will tell you. So what happens if you're following more than one characteristic at the same time? And for our purposes, 2 characteristics is all we're going to be able to handle, because if you get more than 2, it gets really complicated. So we'll just stick to what's known as a dihybrid cross. And in this case, it's going to be a plant with seeds that have a smooth coat and a yellow color, and a plant that has wrinkled coats on the seeds, and they're green. So if you cross them in the F1 generation, they're all going to come out 100% smooth and yellow, like the one parent. Now, if you take from this F1 generation 2 individuals and cross them, and you get a progeny, an F2, you'll find that the majority of them are like the one parent, smooth and yellow. A few are wrinkled and yellow. Another few are smooth and green, and only one is like the green wrinkled parent. So the ratio is 9:3 to 3:1. How do we get to that conclusion? You have to put the gene symbols on And so we know that the smooth yellow one is dominant at both loci. So we call it big S, big Y, big Y. Similarly, the green one, the green wrinkled one, is little s, little y, little y. So the only gametes that the dominant one can make are big S and big Y. And the only gametes that the other one can make are little s and little y. So in the F1 generation, again, you'll have all heterozygotes, big S, big Y, little y, and the phenotype will be all alike, smooth and yellow. But now you ask, what are the gametes that the F1s can produce? Well, it turns out that they can be big S, big Y, big S, little s, big Y, and little s, little y. And so you plot that out on your Punnett square on one axis. And you do the same thing on the other axis. You have all the same genotypes. And then you start asking, what are the combinations? And now you can see why you end up with 9 like the dominant parent and only 1 like the recessive parent. And then there are 3 that are recombinant of each recombinant type. Green and smooth and yellow and wrinkled. So these are the genotypes that come out, and the phenotypes are 9 to 3 to 3 to 1. So do you see what I mean by always asking, what are the gametes that are made by these individuals? And once you know the gametes, you can make a Punnett square for the F2.
So Professor,
Yes.
question. So on the assignment, because I was looking it over real quick, when it comes to answering the questions, do you just want us to explain it, or do you want us to actually do the squares and the crosses? Or
I
do you
want
just
to
want a written explanation?
understand it, which means I think you understand it best if you draw it out. And so this is not a graded assignment. This is for your purposes and learning how to do these things. So my recommendation is that you don't try to just do these in your head, to actually draw them out like you see here, so that you can see the explanation.
Okay.
Are we gonna go over them next class to make sure we got them correct?
Yes, next class, we'll spend as much time as we need going over those 10 questions, 10 problems, and making sure that everybody understands how to do them. Okay, as I said earlier, from Mendel's work, he derived what he called laws, Mendel's laws of inheritance. So the first law is segregation, which says that allele pairs separate during gamete formation and then reunite at random at fertilization. And to visualize this, I refer you back to the last lecture on cell division, where going from anaphase, meiotic anaphase to meiotic telophase, the alleles segregate so that each product of meiosis is different from any of the other products and is different from the parent. That's because the alleles segregate. The companion of that is independent assortment. Chromosomes separate randomly from one another at metaphase. And again, I refer you to this figure from the previous lecture, where the chromosomes line up at the metaphase plate, And each one is going to go to either one side of the cell or the other. And what determines which side it goes to is entirely random. And that's known as either random assortment or independent assortment. And finally, we have the concept of dominance. A dominant allele completely masks the effect of a recessive allele. It produces the same phenotype as either a heterozygote or a homozygote. Now, Mendel was lucky, either very lucky or very crafty, in that he didn't have to deal with levels of dominance, which we know exist. So as we've said, dominance is an allele that masks the phenotype of its alternate. There's also a phenomenon known as codominance, which means that in the F1, you'll see both of the parental phenotypes, discrete parental phenotypes. There's another eventuality called incomplete dominance, in which the heterozygote phenotype is a blend of both the parental phenotypes. And then we're going to talk a bit about sex-linked genes. They're in a special category because of the sex chromosomes. A sex-linked gene means that the gene is on a sex chromosome. So first, we're going to talk about incomplete dominance, where the heterozygote is a blend of both. So to illustrate that, we're going to use an example of chrysanthemums. So this is a real case study. If you take a red one and cross it with a white one, the F1 is neither red nor white, it's pink. And if you cross, if you do go to the F2 and not self, but make a cross between two F1s, you'll find that you have one red, two pinks, and one white. So that's a ratio of 1 to 2 to 1. And this is an unusual situation in which the heterozygote is halfway between the two parents. And the reason I said that Mendel was either lucky or crafty is that each of the 7 traits that he chose for his studies had complete dominance between the alternate alleles. That is, when he looked at progeny, he saw discrete classes. He didn't see a pink coming from a red and a white. So this greatly simplified his interpretation of results. Now, 7 out of 7 is pretty good chances. And so it's commonly thought among geneticists that Mendel probably had other crosses that weren't so nice and clean, and he didn't know how to interpret them, so he ignored them. At any rate, he was right about the ones that he did choose. There's another example of incomplete dominance, this locus that controls a disease, hypercholesterolemia. The homozygous dominant is healthy. It makes LDL receptors. These are cholesterol receptors. And the cell has an adequate supply of them, and it's healthy. In the heterozygote, some receptors are made, but not enough. And so there's a mild disease. In the homozygous recessive, no receptors are made, and there is severe cholesterol disease. Well, let's talk about sex-linked inheritance. So as we discussed before, we all have 44 chromosomes plus sex, males an X and a Y, females 2 X's. So males are capable of producing 2 kinds of sperm, either an X or a Y, whereas female eggs are always X. So if a male X fertilizes an egg, it's going to be a daughter because it will be XX. If a sperm fertilizes an egg, a sperm that is Y fertilizes an egg, it will be XY and a boy. And since the fertilization event itself is totally random, we end up with a population of half girls and half boys. So depending on what kind of a gene you have on the sex chromosome determines what you see in the progeny. If you look on the left here, we have a female with 2 X's, and they both have a dominant allele at a locus called A. So X big A, X big A. The male in this case has the alternate allele on its X, has little a at X, and it has a Y. So this is an X-linked gene. So if in the cross of these 2 individuals, there are 2 possibilities, either an X with a big A and an X with a little a in the girl, or an X with a big A and a Y in the boy. This is a different combination here in the center, where it's X big A, X little a, crossed with X big A, Y. And you can see that now 3/4 of the progeny have the big A allele, and 1/4 are little a, only this pink one here. And then in the right, it's a different combination, X with big A, X with little a, X with little a, Y. And now half the progeny are big A and half are little a. So let's take a specific example, color blindness. Color blindness is an X-linked trait. In other words, there's a gene on the X chromosome that determines that the person is colorblind. So males need only 1 copy of the X chromosome that carries the allele for color blindness in order to be colorblind. Females have to have both copies. So both of the X's have to have the mutation for color blindness. So consequently, fewer females are colorblind than males. So let's take a specific example of a male who is colorblind, and he has children with a woman who is a carrier. So she's not colorblind, but she's got a mutation for color blindness on one of her X's. So what is the likelihood that their children will be colorblind? Well, we can work that out by looking at the gametes. The colorblind male, he's only got 1 X, and to be colorblind, he has to have the mutation on his X. And his other sex chromosome is a Y. So the gametes that he can produce are XB for blindness and Y. Now the female is a carrier, so that means that she has 1 normal X and 1 mutant, XB. So the gametes that she produces are X and XB. You put them into a Punnett square, The female parent can be XB or X. The male parent can be XB or Y. So you get one XB, XB, that will be a colorblind female. You get one X, XB, that would be a normal female, because remember, females have to have a mutation on both in order to be colorblind. So this one can see normally. You have one XBY, which is a colorblind male, because a male only needs to have one mutation on his X to be colorblind. And then the final combination is XY, which is a normal male. So the answer to the question, what's the likelihood that their children will be colorblind, is 50%. Half the children are likely to be colorblind. Half the males are colorblind and half the females are colorblind. And half the progeny are males and half are females. So do you see how to work out a problem on sex-linked genes?
Yes.
Okay. That's good. All right, we're going to do, I think, yeah, one last illustration of a dihybrid cross. We have a true breeding tall purple pea plant crossed with a true breeding short white pea plant. And purple and tall are dominant. What is the genotype and the phenotype of the F1? And then part 2, if you go to an F2, what's the genotype and phenotype of that? Well, I want you to work this out for yourselves. I've done the first part in yellow here, showing you the gametes. So the tall purple parent is big T, big T, big P, little p. All the gametes will be big T, big P. The short white is little t, little t, little p, little p. All its gametes are going to be little p. And so on the F1, it's going to be heterozygous at both loci. And the phenotype then will be all tall and purple, like the one parent. The gametes that are made by these F1s, take two F1s and cross them with each other, the gametes are big T, big P, big T, little p, big P, little t, big, or little p. So you plot those out on a Punnett square, parent 1 and parent 2, and I'll leave it to you to fill in the rest. That's part of your homework. You can do that along with the problem sheet that you have. And finally, a summary. This is again sort of a supplemental study guide for you, for your purposes. First thing I want to emphasize that you know what Mendel did, the two things Mendel did that allowed him to come up with the concept of genes, whereas nobody prior to him ever did. So the two concepts he had were that of inbreeding and developing an F2 generation. Now, in order to think about genetics, you must know the language of genetics. So I have listed here a whole series of terms, and you need to know what each of those is. Some of them are likely to come up on tests, but in order to think about genetics, you really need to know these, whether they're on the test or not. Again, I'll leave this for you as homework. And that is it for today, unless you have questions.
Professor, I have a quick question. So on Monday, there's not going to be any assignment except the practice assignment, right?
Yes, I'll start a new topic if we have time. It may take the entire class period to discuss the problem sets, but that's going to be the main thing on Monday. I want to make sure that you all know how to do those problems. And if we have time, we'll start on some concepts in molecular genetics.
Okay, thanks.
Anything else?
Yeah, Professor, this is Sierra. I just wanted to ask, is capitals always dominant and lowercase alleles always the recessive?
Yes,
Okay,
that's a genetic convention. Applies to all systems.
okay, thank you.
Okay, anything else?
When everyone leaves, can I talk to you one-on-one for just a second?
Sure.
Thanks.
Professor,
Uh-huh.
the only thing due tonight was the worksheet, right?
Yeah, the worksheet with the circles.
Yeah. Okay, cool. This is Katrina Vebert.
Okay,
Thank you so much.
okay.
Have a great day.
All right, thank you.
Is that okay to leave right now?
Sure.
Okay, thank you. Have a good one.
Okay,
Thank you.
all right. Okay, I'm gonna stop sharing. All right, if there's nothing else, I will end the session and see you all on Monday.